Hello friends, today we'll be talking about a protein called as P53. So P53 is also known as tumor suppressor protein. Okay, so as the name suggests, these are the protein which suppresses the tumor present within the cell. Okay, so let's see how this works. Suppose there is a DNA here within the cell, and because of some external factors like UV radiation, this DNA has gone some mutation in this part of the DNA. Okay, so now if these kind of DNA replicate further within the cell, those cells become the tumor cells. They become the tumor cells causing the cancer to the cancer to the body. Okay, so it is very important that this kind of DNA has to be stopped from replicating within the cell so that they don't spread the tumor in the body. Okay, to see how this kind of DNA is stopped from replicating within the cell, we'll understand the cell cycle first. This is the cell cycle, suppose, and you have the G1 phase, S phase, G2 phase, M phase, and the G1 phase again. So this part of the cell cycle is known as the synthesis phase. This is the phase of the cycle where the parent DNA is replicated into the daughter DNA, like they are doubled in their number. This is the phase where this kind of DNA are replicated within the cell. So it is very important to stop the spread of these tumor cells, to stop their movement from G1 phase to the S phase of the cell cycle, so that these kind of DNA are not replicated further in the cell. So how does this happen? So they are different checkpoints within the cell. There is a checkpoint present here, there is a checkpoint present here, and another checkpoint is here. Okay, to enter from one phase of the cell cycle to the another phase, the cell needs permission from a complex called as the CDK and the cyclin. This is a cyclin CDK complex. To enter from this phase to this phase of the cell cycle, the cell will take permission from the CDK cyclin complex. Okay. Now to stop the production of this DNA from in the S phase, this cyclin CDK complex has to be manipulated so that these complex do not give the permission to the cell to enter from G1 phase to the S phase of the cell cycle. Now, so let's see how this happens. Now, a protein will detect that there is a mutation within the cell, and that protein in turn will send the signal to the P53 protein. These P53 protein will then go and bind to a gene called as WAF1 gene. Here the P53 goes and bind to this gene. Now this gene induces the production of another what we call protein called as the P21 protein. These P21 will again go and bind to the CDK cyclin complex and gives a signal to the cell. Now this cyclin CDK complex will give the signal to the cell that you cannot go from G1 to the S phase of the cell cycle. So the cell cycle is halted at this phase of the cell cycle. It cannot undergo from G1 phase to the S phase of the cell cycle. In this way, the mutation is detected here. Then this is sensed by a protein, and that protein sends a signal to the p53, and that p53 goes and binds to the WAF gene in the cell. This WAF gene produces a protein called as a p21 protein. This p21 protein goes and binds to the cyclin CDK complex, which gives the permission to the cell to go from one phase of the cell cycle to another phase of the cell cycle. And this when p21 binds to this complex, the cell cycle is halted at this stage of the cell cycle. And this p53 also produces the transcription factors which enables the repairing of this DNA. And even after this, if the DNA is not what we call repaired, this cell undergoes a apoptosis, which is the programmed cell death. The cell goes through a programmed cell death. The cell dies within the cell so that it cannot spread the tumor in this body now. So it is seen that by the research, the patients with cancer have the mutant p53 protein. Okay, so in the body, in the normal cells of the body, the p53 levels are very low because they are continuously degraded by a protein called as MDM2. Because they are not needed in the body, there is no need of this protein, so these proteins are degraded by MDM2 protein. Okay, these are the degraded p53. This is the MDM2. And now when a cancer or a tumor is detected within the body, now this p53 has to grow in number and do its all of its function, binding to the WAF1 and producing p21, and then binding to and then p21 binding to cyclin CDK complex and halting the synthesis of this DNA at the synthesis phase. Okay, so to happen that this MDM2 complex should not bind to the p53 so that it's not degraded. Okay, so what then happens? This p53, sorry, this p53 then is phosphorylated. When this p53 gets phosphorylated, the MDM2 cannot bind to the this cannot bind to the p53, and it does its function now. The p53 is now active. This is the phosphorylated p53 and it does its function now. Here it is not degraded. Okay. In this way, in the cancerous patient who are developing the tumors within the body, in their body, the p53 levels are seen to be high. And in the normal patients, this is in the normal patients, normal people, the p53 levels are very low because they are continuously degraded. And in the tumor patient, in the cancer patients, their levels are seen to be high because they have to perform their function to stop the production of this malignant DNA. So hope you've understood. Thank you.